Welcome everybody to the afternoon session on structural and functional brain networks. In the interest of time, we start immediately with Eve Marder from Brandeis University. The title of her talk is Differential Resilience to Perturbation of Circuits with Similar Performance.
I'd like to thank everybody for inviting me, and Nikos, congratulations, and it's a great pleasure to see you. I saw him and I disappeared, he's disappeared again. But it really is a tremendous honor to be part of this symposium. What I wanted to do today is just show you a snippet of the problem that has been most preoccupying me, which is, for the last 5 or 8 years, which is to try and understand two things related to individual variability, and that is to say how sensitive circuit performance is to a variation or variability in underlying circuit components, and then to see the relationship between that and resilience to perturbation. And I will introduce that as we go. But before I do that, I just want to honor the people who've done the work. My present lab is up there, some really wonderful people. My lab is usually, and now, about 50% experimentalists and about 50% theorists, and they do talk with each other, which is always nice. And then I have a number of wonderful lab alumni, where some of them are postdocs elsewhere, etc. And then some number of people have gone on to industry and government, and then a large number of people have gone on to run their own labs. And I like to show this slide in the beginning to encourage all the people who are in what I like to call postdoc purgatory, which is this holding pattern as you sort of wait to assume your final career path, and to say, first of all, there is hope, and secondly, there are many, many different wonderful paths. And I'm very proud of them. And I'd like to say one more thing, which is Jaksu Ki, who was up there under my mentor, has recently died, and I miss her a lot. And I'm sorry that you all didn't get to know her. She's really a wonderful scientist and person. So these are the questions that I'm going to be addressing. How tightly tuned do the parameters that govern synaptic strength and intrinsic properties need to be for good enough circuit behavior? Or how different are the underlying structures of normal, healthy brains? And I often say at this point that we as scientists grew up sort of measuring means and standard errors and pretending that every individual was a representative of the Platonic ideal of the mean. But on the other hand, every 2-year-old knows that every individual person is different from every other individual. So I'm trying to bring you guys all back to what you knew when you were 2 years old, which is to revel in the fact that we are all different. And then the key question, I think, for us going forward is, how is individual variability related to resilience to challenge? Because this is one of the problems that our animals face in this area of climate change, and I've been pushed to studying that. And it's how we behave as we confront an increasingly complex world with many stresses. And then the last problem that we've been studying a lot, but I probably won't have time to address, is how is stable network function maintained over the animal's life while allowing for adaptive change. So these are questions that I'm going to address using the crustacean stomatogastric nervous system. This comes from lobsters and crabs, and I realize I'm probably the only model system here. I'm not a model system, but maybe I am a model system, you never know. But I think these same questions are equally relevant to the human, monkey, mouse brains as they are to lobsters and crabs, where we're going to be studying it. Now, the system that we use is, as I said, the crustacean stomatogastric nervous system, and the network I'm going to be largely discussing is the pyloric network of that system. So just very briefly, for those of you who didn't grow up in the old world, what we do is we buy lobsters and crabs from distributors who buy them from the fishermen, who of course lie about where they got them, because fishermen always lie about where they catch their animals, right? And then We bring them into the lab. They actually take a taxi from the waterfront to Brandeis. We put them in seawater tanks. We hold them for a little while, and then we take an animal. We dissect the stomach out of the animal, and then dissect the nervous system off the stomach and put it in a silver-lined petri dish that we then can superfuse with physiological solution of any composition that we want. And when we do that, the pyloric rhythm, which is seen here, will last for days, depending, certainly for many, many hours, but often for 5 or 6 days, as long as we keep too many bugs from growing. And it continues to produce this rhythm motor pattern that we call the pyloric rhythm. It's a triphasic motor pattern with a constrictor 1, constrictor 2 dilator, constrictor 1, constrictor 2 dilator phase, where the LP and the PY are the constrictor 1 and constrictor 2. The PD is the dilator, and these motor neurons innervate muscles that open and close a valve at the bottom of the animal's stomach. And these are now simultaneous intracellular recordings showing a depolarization, a burst of action potentials, a hyperpolarization, etc. And you can see that triphasic rhythm down here on the extracellular recordings, LP, PY, PD, LP, PY, PD. And I should say these neurons are unambiguously identified on the basis of which muscles they innervate. So one of the advantages of this system is we have unambiguous cell identification. Another advantage is that the system that we have in vitro, these fictive motor patterns, are almost identical to the motor patterns that we record in vivo. So we can actually do experiments in vitro knowing that the ganglion is basically operating by some of the same mechanisms that it would in the animal. Now, this is a connectivity diagram. Uh-oh. Oh, this thing. This is a device of the devil. No, that's why it's a device of the devil. There's the devil. But I don't have the dongle in, so why is it doing it? He's the only person over 40 I've ever known to be able to use one of these things. How do I know he's over 40? I know he's over 40 because I've known him for a long time, but it shows how young at heart he is. But anyway, that's why I don't use them, because you see, this is the connectivity diagram that gives rise to these motor patterns. And in my connectivity diagrams, if I have them, the resistor symbol means electrical coupling. These filled dark circles are chemical inhibitory synapses. So by 1980, we had this connectivity diagram, and we had our first word model of how this preparation worked. Now, I'd like to say, before going any further, this today would be called a connectome. And you also know that if I showed you this connectome and didn't show you these dynamics, there's absolutely no way you'd be able to go from this to this, because too much information is missing. And what's missing are the identification of the voltage and time-dependent currents in each of the cells, their conductance densities, and the strength and time course of all these synaptic connections. And then even if you did what we did over many years, which is to measure in voltage clamp all of those components, you'd still need some way of putting that information together to basically go from the analysis of single components to the system dynamics. And that's why, as a perfectly good experimentalist as I was, ended up working with computational people just to build models to allow you to put back together the insights you get from the reductionist perspective. So that's how I ended up working with theorists. And I'd just like to make one more comment in passing. One of the reasons why I still work on this nervous system is because as a central pattern generator or a motor system, it has a very easily measured motor output, so therefore we always know when it's doing the right thing, when we've perturbed the output, and that motor pattern is exactly what matters for the animal. Okay, so a number of years ago, Astrid Prince, who was then a postdoc in my lab, wanted to build a model of the pyloric rhythm, and I said, fine. And So she set out to do that, and she started out with a model of a single neuron that we had, and she was German, she's probably still German, and she sat at her desk and she was saying really bad things. Now, I don't understand German, but I knew these were bad things. And then all of a sudden she went silent, and I sort of went by and I said, "What's going on?" I asked her. She said, "I solved it." And I said, "What do you mean?" So she had been trying to hand-tune this model, and that's the route to madness, by the way, of anybody who tries to hand-tune a model. So she decided to take a single neuron model that had 8 different kinds of conductances, and then she chose 6 values for each of the 8, and she simulated 1.7 million model neurons, and then she selected from them neurons that had the behaviors she was looking at. So this was really a shift in the way how to get successful models. So doing that, she found a number of models for that pacemaker ensemble that I showed you before, and a model for the LP and a model for the PY. And then she chose either 5 or 6 values for the synaptic connections, and then she simulated all against all again, and she simulated 20 million model networks, and then she asked which ones of those simulated models were good models for the pyloric rhythm. And so here's all her networks, her 20 million. 19% of them were pyloric-like. That means they were triphasic in the right order. And that was LP, PY, PD. And then 2.4% of them were good models of the pyloric rhythm in that they had behaviors that matched the range of behaviors that Dirk Bucher had measured in 99 animals. So Dirk had measured the frequency of the period, the number of spikes per burst, the phase relationships, and a number of other criteria. And Astor just asked for a model to fit within all of those distributions. So what was really important here is instead of trying to build a model to capture mean behavior, she was looking for a family of models that would capture the range of activities we normally resulted from. And so then we'd end up with X number of models that would be a good model of the range of the physiological behaviors. So, and there were 400,000 of them since she started out with 20 million. So here's what 2 of them look like. Model 1, Model 2. You probably have, many of you have seen this slide before. And when you now look at their underlying conductances, you see that even though these 2 model networks have very similar performance, their underlying parameters are very different. This is the H conductance, that's IH, that's the hyperpolarization activated inward current, and the PY, here it's big, here it's small. The PY to LP synapse, here it's big, here it's small. The ABLP synapse, here it's big, here it's small. You can see this set of parameters and this set of parameters are entirely different, and yet the total result is a very similar motor pattern. Now, when I saw this originally, I was incredibly happy because, first of all, it was really interesting, and second of all, it made me feel better about the variation that we were recording in our biological systems. Because as an experimentalist, you always worry about how much of the variability in your data is your fault and how much of it's real in the population. And this gave us belief that a lot of the variability we were seeing was actually real. And you'll see in a moment, what we and others see is about a 2 to 6-fold range of variability in almost any parameter like conductance density or synaptic strength across animals. And that, I think, is actually a really important number because it's something that biological systems can achieve, and yet it's a good enough, creates enough of a set of constraints that you can get good enough behavior from it. We can come back to that. So on the basis of this, now when I first started talking about this, there were these pesky physicists in the room, and they had a lot of bad things to say, but like they told me this couldn't be right because biological systems were optimal. And I said, well, you're not optimal, so why should my crab be optimal? But leaving aside that, the one thing they did say that was reasonable was that even though these 2 behaviors were similar unperturbed, that there was bound to be a perturbation that would separate these, because the underlying parameters are different, and that's absolutely true. There has to be a perturbation in the universe of all possible perturbations that would separate these. And so then that got me thinking, and I realized that we had a really interesting possibility to say, well, if my crabs have this kind of variability across them, then the question is, can they nonetheless respond reliably to the perturbations that are relevant in their life, even though there is almost certainly likely to be a perturbation that will separate them? So we set out to answer a few questions. How variable are the parameters in real biological networks? Do perturbations reveal cryptic animal-animal differences? And then how reliably can animals with intrinsic variability respond to perturbations? Now, I think I'm going to quickly skip through this slide and go to this, which says that we've done a lot of voltage clamp measurements and a lot of mRNA measurements, and the answer is that individual synaptic intrinsic parameters can vary 2 to 6-fold across animals, even when they have very similar behavior. And if you unpack most people's biophysical data, that's the number you get, that 2 to 6-fold range. Despite the fact that if you look, and these are recordings from 5 animals, the recordings are very, very similar despite this 2 to 6-fold variability in the potassium current density or the sodium current density or whatever. So then we said, what happens if we start looking at perturbations? And we were here looking for global perturbations, perturbations that would be likely to influence all cells and all properties of the network. And over the years, we've studied temperature, which is really a global perturbation that will act on every single biological molecule or set of molecules, but differentially, and we'll talk about that in a moment. We've also studied pH. We've also studied high potassium. It's a perturbation that many experimentalists use. But I'm going to only focus on our data with temperature because in some ways it's the most interesting and it's the most relevant to our current world. So, temperature. If you think about that network, that pyloric rhythm, and if you think about the fact that all of the timing and the phase relations to the network depend on membrane currents that basically have fixed time constants. The rates of opening and closing of ion channels have rate constants associated with them. And if each one of them is temperature sensitive but differently, you might imagine that they can get out of whack with each other. So the problem of temperature regulation becomes actually quite challenging. So biologists often define the concept of a Q10. There's the equation. A Q10 of 1 means it's completely temperature compensated. A Q10 of 2, that's what I was taught when I was a child. Network biological systems doubled with a 10-degree change in temperature. Most ion channels and similar things have Q10s between 2 and 4. But there are some channels that have Q10s at 50 or 100. They're very, very temperature sensitive. And I'd just like to show you one slide that describes why this is such a problem for an animal that has to deal with changes in temperature, such as you do. So this happens to be a simulation that Tim O'Leary made. And he started out with a bursting neuron. And here it is at 10 degrees. And then he used 3 different sets of Q10s for the voltage and time-dependent conductances in the model. And then as he changed temperature, this one went from being a burster to being a single spike burster to a depolarization block, whereas this one, when you went cold, went into a single spike bursting mode, and when you go warm, it went into a tonic firing mode, and then it died in a hyperpolarized membrane potential. And this one then went slower and then faster, but eventually started firing tonically. And that's because even though the currents worked well together at 10 degrees, as the different sets of Q10s pushed and pulled on all those different currents, they no longer were able to maintain bursting. But what about the crab? So these are data that Lamont Tang generated. So here's the pyloric rhythm, PD, LP, PY at 7 degrees. Here you have it at 19 degrees. PD, LP, PY, beautiful triphasic rhythm. And then when Lamont plotted, this is a semi-log plot, he had an apparent Q10 of about 2.3 for the frequency of the period. So the rhythm gets faster, but, and this was astonishing when I first saw it, the phase relationship says to say the normalized time in which each cell fired was completely constant. So that if you think about it, for me to walk, if I walk slowly or I walk quickly, I still have to get the right pattern of extensor and flexor activity and left-right alternation in order to walk normally. And so here, this triphasic rhythm is able to maintain its pattern as its frequency is changing. And so this, if you think about it hard, you'll realize it's actually quite a difficult problem. And then here are the same quartz sorts of data with intracellular recordings, 7, 11, 15, 19, 23. You can see the rhythm gets faster, but remarkably, the waveforms, if you scale out time and superimpose them, are remarkably maintained over that temperature. So what Lamont found in these first set of experiments, that from about 4 degrees to about 23 degrees, these networks were perfectly temperature compensated. On the other hand, when he went higher in temperature, and now we're looking for more extreme perturbation, a perturbation which takes the system beyond where the ocean should actually ever let them see. And we'll come back to that. He started seeing what he called crashes, and every animal showed a different kind of crash. That's disordered activity with interrupted or irregular bursting, or some units going silent, others firing tonically, etc. And then if you go still higher temperatures, Lamont found that here he had a perfectly good rhythm at 23. At 31 degrees, it was completely crashed, but then when he went back to 31 to 23, it came back. So what Lamont actually articulated at the time is that temperature crashes are widely variable within the population, likely because of animal-animal variability in underlying circuit structures. So, but every animal we've ever looked at responds really well to temperature in its permissive range, and you only see the influence of those different membrane conductances when you go from the permissive range to more extreme perturbations. So, now I forgot, is this going to be green until when? Okay, well, I know, but okay. So then the question is, our system is modulated by large numbers of different substances, and so we wanted to ask whether altering the modulatory environment would alter crash sensitivity, susceptibility. So the first thing I'd like to show you, and these are, is if you leave the modulators on, and everything Lamont did was with the modulators on, you get that beautiful pyloric rhythm that is phase constant. Here you see it, an average period of about 1 hertz. However, if you remove those modulatory inputs, now the network slows down and becomes much less regular, and you actually lose that phase constancy because you can't maintain it at very low frequencies. So, and you actually see that the difference is this tremendous amount of animal-animal variability in the motor patterns are revealed when you remove modulators. It's as if the modulators have been hiding the amount of variability that's there. And so this is, for example, animal 1, as Sarah Haddad increased the temperature. Animal 2, animal 3. Animal 3 started out with a lousy rhythm and got worse. Animal 1 started out with a pretty good rhythm and stayed pretty good. And animal 2 was in between. So the first result was that removing the intrinsic descending modulators revealed substantial animal-animal variability, which was hidden before that point. So Sarah came up with a way of classifying these disordered patterns. So in purple, you see the normal triphasic rhythm, faster than about 0.8 hertz. Yellow is a really slow triphasic rhythm. Then you have all sorts of other colors with all different kinds of patterns. And so then she was able to go through the data. The 3 animals I showed you before, the animal 1 started out slow and just got faster, so it went from yellow to purple. Animal 2 started out slow, got faster, but then at high temperatures started breaking up into other kinds of rhythms. Animal 3 started out lousy and then went through some decent triphasic rhythms at high temperatures, but was basically intermittent. So then we wanted to do, or Sarah wanted to look at the effects of applying modulators to the temperature sensitivity. And so she's going to look at 3 different modulators. One is oxytremurin, which you know is a muscarinic cholinergic agonist. 2 is a peptide called proctolin, which activates the same current that muscarinic agonists activate. And it's a voltage-sensitive inward current that has an IV curve very much like the NMDA receptor. So it sustains oscillations. And the 3rd was serotonin, which we expected would make a mess, and it does. Okay, so here's 2 animals in oxytremurin. In the presence of oxytremurin, you can see the rhythms are strong, and as you increase in temperature, you can see the activity is maintained over the whole temperature range. It's going faster and faster. Proctolin produces some pretty slow rhythms at 11 degrees, and you get increases in frequency, but you maintain the rhythm. And that you can see here in these color blocks. You do fine in oxytremurin and proctolin. However, in serotonin, the control data are incredibly variable. And now as you increase temperature, you see all sorts of disordered rhythms. And that's seen here in the color blocks where this animal started out okay and then got worse. This one started out okay for a short period of time and got worse, and you can see all these different transitions between states. So just to summarize these data, at low temperatures, all networks are well behaved, showing consistent changes in frequency and outstanding temperature compensation. This is important. But each individual animal crashes with individual dynamics as predicted by their underlying parameter differences, and some modulators may expand the stable range of temperatures, while others make the preparations more temperature sensitive. And you can see there was a tremendous amount of animal variability in the effects of serotonin. So this was all to lead up to talk to you about finding models that can do this kind of temperature compensation. As I said before, it's a very challenging problem because to get something that will work well at 2 or 3 or 12 different temperatures, you have to somehow or other figure out how you're going to compensate for the fact that all the Q10s of the components are going to be different. So you have to figure out how you find a path through that. So the question is, how do you find models that can do this? Because we know the crabs can do it. And then you might also ask, how might biological systems find neurons and networks that are well temperature compensated? Because one of the first things we learned in building models is that the solutions that can do temperature compensation are very, very rare in randomly sampled parameter space. So I want you to remember that. We found a way, Leandro has a way of finding the solutions, but they're very uncommon, and you wouldn't find them randomly. And then Tim O'Leary built a model that finds them, and so we can talk about how you can find those paths. But let's talk about Leandro's data first. So what Leandro did is he built a pretty smart genetic algorithm that actually allowed him to find pyloric rhythms that in the first instance were temperature compensated. And I'm going to skip this, but show you the way we're going to visualize these data. So in the top you have the voltage trace of voltage as a function of time. And now what we're going to do is display the underlying currents as a function of time that give rise to this voltage waveform. So the top black, this is a log scale. We're displaying the total outward current and you can see the total outward current is higher here as you have spiking. And down here in black, again on the log scale, the total inward current. And then in the colors, we're displaying the currents of each of the currents in the model. This model has a sodium current, 2 calcium currents, 3 potassium currents, an H current, that's a hyperpolarization activated inward current, a leak, and a synaptic current. Okay, so if you just look at this, you can see that early in the burst, the purple is very small, it gets a little bit bigger, it gets a little bigger. Early in the burst, the orange, which is the calcium-activated potassium current, starts out pretty small, it gets bigger, and then at the end of the burst, it gets very, very large, etc. So the next thing you're going to be asking me to show you, hopefully, if you're awake, is what happens if I look at 6 models that have very, very similar behavior, but now we're built with different underlying conductances, like Astrid did. What can we see in the actual currents? So if we just look at the purple, that's the A current, that's the transient outward potassium current. Here, the A current is doing almost nothing. It's very small. Here, it's doing more. Down here, the A current is a massive contributor to the dynamics of the neuron. So this neuron and this neuron, although they have very similar behavior, the A current is far more important in this one than it is in this one. And likewise, the calcium-activated K is bigger here than it is here, etc. And you could spend a long time looking at those. So this just allows you to see that there are different ways that these currents can work together to produce these dynamics. So then Leandro added a term, and now he asked his algorithm to find him networks that were temperature compensated up to 25 degrees, and they would crash by 30 degrees. So here's one of them. Here's the rhythm at 10 degrees, 15 degrees, 20 degrees, 25 degrees. You can see the rhythm is maintained, and then here it's crashed at 25. And then, because he had in his mind Lamontagne's original data on phase, this is just the same recordings I've just shown you, but now he's plotted the phase relationships of the starting and stopping of each of the neurons in the cycle, and you can see those are phase constant across temperature from 10 to 25 degrees. So he's actually found models that replicate this very difficult to find, really intriguing behavior. But now that since we have these, we can ask a question, what can you learn about how these models work? And I'm going to show you, which is a slide that's become my favorite slide, and I call it tiling time, because I think it has a really deep insight into what's going on. So what Leandro's doing here, he's just showing you the last spike in a burst for 3 different models at temperatures that go from 10, 11, 12, 13, 14, up to 25 degrees. So we're going to look at the currents following the last spike of the burst in these 3 models. And we can actually start with the second model here. If you look at this last spike, there's a pretty big purple blob, which now decreases as you get warmer. But the orange, which is the calcium-activated K, is getting larger and larger and larger as you get warmer. So this cell is transitioning. The mechanism of spike repolarization is changing gradually as you move through temperature. At the other hand, a similar result, that is to say the currents change over temperature, is seen in these other 2 models, where here, again, the purple gets smaller and the orange gets bigger, but the orange gets less bigger here than here, but the yellow gets increasingly small as you go. So these are 3 different models, which all do what I mean tile time, which means they slide gracefully and gradually through the transition as temperature is changing. They basically slide through the temperature transitions, the mechanism. And I'm almost done, but I'm going to show you one more. This might be Leandro's favorite slide. I don't know if it's his favorite slide, but he likes it. So now we have 2 models, model 1, model 2, and now we're just looking at the currents. So here's the voltage waveform, and now here we have the sodium current. It's small at 10 degrees, it's big at 25 degrees. And the other model, the sodium current was big at 10, is getting smaller at 25. As we go down, one of the calcium currents is big at 10, gets smaller at 25, whereas in the other one, it gets bigger at 25. Here, the other calcium current is small at 10, gets big at 25. This one starts out big and gets smaller as you go higher in temperature. So here again, we're seeing smooth transitions in behavior, in mechanism, but the important thing is that because you have so many currents, you can slide through these different mechanisms. And these two neurons, which have very similar behavior, are doing it mechanistically differently. Now, we're going to build 3 networks, and I'm going to be done, which have different underlying conductances. So this is network 1, network 2, and network 3. And there's the red, the green, and the blue. And we have all those currents. And I'm just going to show you the red network crashes over here. The blue one, the green one crashes a little bit differently, and the blue one crashes still differently. And I'm just going to very rapidly go back and forth between these 3. Just want you to look at the last panel with your Gestalt viewer. There's the red, green, and blue. And you can see the crashes are also qualitatively differently. And I will skip the next slide, although it's cool, and just finish up and say that individuals with similar starting circuit performance with relatively similar circuit mechanisms in the control system can nonetheless show entirely different crash mechanisms. And this is a cautionary tale for thinking about disease or thinking about how any of us deal with perturbation, because it says that you could have a whole population which is perfectly fine in the natural order of things, and then the ocean warms up and my crabs flip out, and you give people the same drug, and 85% of them do fine, and 15% of them do really, really badly. And it's for exactly the fact that they're starting out with different solutions that are, in the unperturbed state, are all benign. So stop there. You have to give him the mic. I don't need another mic.
She already has a microphone. So I've watched this story evolve, and it used to be millions and millions, and they were all fine. And now you add constraints. You add neuromodulator response. You add temperature response. And as you add new constraints, the number of available parameter solutions is decreased. Hello?
Yeah. So the question you're really asking, I think.
The question is, where does that converge to? I mean, what's the, there's some variability from individual to individual, I understand that, but is there in fact really just is there a set of a few basic states which really account for all of the variables you can impose on the system, or is it going to be as diverse as you imagined from the beginning?
I think it's still going to be quite diverse, but what we're doing now is we're looking at combinations of perturbations to ask whether, if you change pH and temperature at the same time, do they interact? And so that's what we're doing right now. You still have many different individuals that persist, but you can see that there is an interaction. If you have 2 things, both of which are bad at the extreme, they can interact. But some animals will do remarkably well, even with the 2, and others are more defeated by them. Now, the critical thing is, the other part of your question is, we don't exactly know whether temperature, pH, oxygen levels, a variety of other things, the tide, moons, what's most important for the life of the crab. So we've looked at 4 or 5 perturbations and their interactions, and that's where it's heading. There.
Does the crab have a temperature-sensitive neuromodulatory system to do what you did in the lab?
Presumably, it probably has something that would work sort of like that, because there are 35 to 50 modulators. Many of them are circulating hormones, and many of the descending modulatory pathways are activated by sensory feedback. So it's very likely that some modulators could be released in response to extreme temperature, which could then function to buffer the animals to expand if they expand their operating range. And my guess is that's partially what's going on. But nonetheless, eventually you'll still run into crash.
Thank you, Eve. Beautiful. You may have begun to get at this in the very last half of a sentence. You talked about crashes that could, if you change the temperature back, they could be reversed. But do you find that there are some that are irreversible in a relatively short space of one of the things like temperature or pH, so that we could expect, as people say, 2 degrees is about enough to have us go off the cliff?
Well, we didn't talk about acclimation, long-term acclimation.
That's what I mean.
These animals show long-term temperature acclimation, so
It
that
doesn't necessarily mean that a crash is a long-term crash.
no. So I was showing you only acute data right now.
Okay.
But if you take the animals and put them in the ocean, in an ocean that's 6 degrees too warm, what we do, and that happened naturally, is the crash points shift up.
Okay.
Because of long-term acclimation. On the other hand, if you go too warm for too long, you produce death.
Right.
Right.
You should tell this to the boss in the White House.
Our boss in the White House does not listen to the likes of me. But I'm going to talk to congressional staffers next week. So I'll do my best.
Thank you so much for your talk. So one of the things that I observe is that you have very similar outputs, very similar spiking outputs, but a very different interaction between all of the different factors that you put in your model. So my question is, do you think that although we see these spikes, maybe the information that they are transmitting is different? Would you think that the spiking pattern does not only count, but also the interaction that happens inside? Do you think that this might store a different language?
I'm a firm believer in that biology matters. On the other hand, people who only record extracellularly don't know. So I think that's the problem for the systems neuroscientists in the room who only measure spikes, is you can see similar spiking patterns and they could be arising from completely different circuit mechanisms. And I would imagine that they do, especially in a well-trained monkey. Okay.
I was wondering, do you see a number of different patterns emerge, because the patterns that emerge and when they're put under stress doesn't kill the animal, but so it allows different adaptations to develop. Had it been that if it got out of range, it killed the animal, it might lead to a narrowing of the number of permutations that are useful.
Right, so it's a little tricky. Remember, I'm working with wild-type animals that are 5 years old, so they've been through a lot of misery in the North Atlantic Ocean. So I'm looking at the survivors. I'm looking at the animals that are more resilient. In the summer, just to go back to when Anne was talking about, you were talking about, the summer of 2012, we saw animals that produced motor patterns faster than we'd ever seen in our lives. And that was the summer that was the winter, the summer after the winter, where the ocean was 6 degrees warmer than it ever should have been, it ever had been. And we saw it in our laboratory animals. They weren't crashing until 33 and 34 degrees. And we never knew whether all the sensitive ones had died, and we were seeing the survivors, or whether our animals had gone north to Maine, along with the squid and the dogfish, and whether we were seeing Long Island animals that had come north, or they had gone deeper. It's really unclear. But we see in our laboratory animals and their crash behavior, we see the traces of the temperature history of the last 6 months. So we keep track of everything now, and you see these yearly cycles. But it's very frightening to see in the lab the effects of climate change.
So thank you very much.